Okay, let's try to understand now the physiology of hearing, and specifically we need to understand how the cochlea is converting the vibrations into action potential that will be transported to the brain, to the auditory center of the brain. In order to do this, let's go to do some drawing. So we said we have the cochlea, and the cochlea, sometimes we call it the spiral organ, it's like a snail shell. So it wraps around itself almost 2 times and a half. Okay? So this wrapping would create, remember this is like a bony labyrinth. So it will create a tunnel. Now, if I extend this tunnel, it will appear like this. So this is the cochlea after extending. And inside what we will find, remember that we have the bony labyrinth and we have the perilymph. So inside we will find a sac that contains the perilymph, which means we have 3 regions now. This is 1, this is 2, and this is 3. The first and the third regions are open or continuous at the peak or at the tip. This area here is this area. So if you go from here inside, you go inside, all the way inside, you can turn back. You can come and leave. Okay? And for a reference, the oval window is somewhere here, and the stapes will be laying over it. But now let's see those 3 regions. What are they? The first region, which is this one here, we call it the sacala vestibuli, and the lower one we call it the sacala tympani. So the sacala vestibuli and the sacala tympani are continuous or connected to each other. Inside we will have the sacala media, or sometimes we'll call it the cochlear duct. Okay, now the membrane that separates the sacala vestibuli from the sacala media, this membrane we will call it the vestibular membrane, and the membrane that separates the sacala media from the sacala tympani, this we'll call it the basilar membrane. Okay, and also just to make sure that you are aware of this, although we're not drawing, here is all filled with lymph or the perilymph. Okay, and also here, although it's a different type of lymph, that's why I'm putting it different color, but still it's all filled with lymph, all filled with the fluid. So the vibrations, when they are coming from the oval window against the stapes, so we are creating vibrations in those fluids. Okay, and we will come back to those vibrations, but just to let you understand, at least for now, that we have a fluid. Those are not empty spaces. No, they are filled with the fluid. Okay, so I'll delete all of this because there are other details that we still need to see before we try to understand what's going on within the sacala media. So we said this is the vestibular membrane. What does this mean? This means if you are standing here and you are looking up, you will see the vestibular membrane above you. And if you are looking down, you will see the basilar membrane. Now, if I made a cut here in the spiral organ or in the cochlea when it's rubbing against each other, I will see the 3 chambers. They will appear like this, something like this. So here, this one will be the sacala vestibuli, this will be the sacala tympani, and here is the cochlear duct or the sacala media. So they are sitting above each other. So this is like a side, this here is a side view. This is if we stand and look like a direct view, or anterior view if you want, anterior view. Okay, those are the 3 regions that are forming the cochlea. Now, inside the sacala media, we have a group of cells. Those we'll call them hair cells. Why do we call them hair cells? Because they have extensions or hairs coming out of them. So those are the hair cells. Hair cells. Here, if I want to draw them, I will draw them all over the floor of the cochlear duct. They're all over the floor. So technically the floor is covered with those hair cells. They have the little hairs. Okay? Now, from the basilar membrane, remember this is the basilar membrane. The basilar membrane is the floor of the cochlear media and the roof for the sacala tympani. From the basilar membrane, there will be another membrane coming toward those hair cells. And technically it touches those hair cells. This membrane, we will call it the tectorial membrane. Think of it as a shed, okay? Like a shed or umbrella above those hair cells. If I want to draw this tectorial membrane here on this drawing, it will be something like that. Also continuous membrane that's touching the hair of those cells. Okay? So that is the situation inside the sacala media. Now, this, I will remove this little guy. I will remove all of this because we will see some additional details about this. But for now, this setting of the hair cells and the tectorial membrane, we call it the organ of Corti, the organ of Corti. And what is the function of the organ of Corti? It converts vibrations into action potentials. So those hair cells, the hair cells are the one technically convert the vibrations into action potentials. Those action potentials will be transmitted to the brain through vestibular, or in this case, like the cochlear nerve, which is part of the vestibulocochlear nerve, the cranial nerve number 8. So here, those will have their own axons, okay, that will connect to the cochlear nerve, okay. So that is the setting inside the cochlear duct. We have a group of cells. Those cells, they have hair. Those hairs are touching, or above them there is this tectorial membrane. It touches them, okay, and we have the sacala vestibule, the sacala media, and the sacala tympani. All of them are filled with the fluid, right? And we said the function of the organ of Corti is to convert vibrations into action potential. So what we need to understand now, how this process is happening. And to do this, I will draw again the cochlear duct, okay, and I will have here the sacala media, okay, and I will draw this line as the tectorial membrane, okay, and I will draw this line as the organ of Corti, and they are touching each other. Now, this line, if I look at this line from above, what I will find it like this. The tectorial membrane or the basilar membrane even, those are technically are like fibers. So here at the bottom of the cochlea, those fibers are short. And as we go to the tip of the cochlea, those fibers will get longer. So those are like this. Fibers. Okay? As we move toward the tip of the cochlea, those fibers will get really long. Whereas at the base, they are extremely short. Now, those fibers, you can think of them those are of the basilar membrane, okay? Or you can think of them of the vestibular membrane. It doesn't matter really which one are they, because we're talking about the same concept in both of them. But what is more important than this is to understand that those fibers will bend in response to vibrations, okay? And those vibrations now they are coming in form waves in fluid, in the lymph or in the perilymph. The bottom or the base, or yes, the base or the bottom of the basilar membrane would respond to high vibrations, whereas the base or the tip will respond to low vibrations. High vibrations usually as a result, or they are coming because we have high frequencies or high pitches of sounds. Sounds with high pitches, they will produce high vibrations. Whereas low vibration, it's the low sounds, the quiet sounds. So the quiet sounds cannot vibrate those fibers. No, the quiet sound will travel all the way to the tip of the cochlea, and it will cause those vibrations or those fibers to vibrate. I usually think of it as small bushes, if you have small bushes, okay, and if you have a long tree or a big tree, okay. So if there is a breeze, the big tree, usually the leaves in the tree or those the branches will go along with the breeze. Whereas the bushes, they need much stronger wind to move them or to bend them, right? So the shorter fibers would need high frequencies to bend them, whereas the longer fibers will need low frequencies to bend them, okay? So now back to here. We understand that the vibrations are coming from here. I'll remove those arrows. The vibrations are coming from here, and those vibrations would run along the sacala vestibule. Now, for each one of those vibrations, it has a certain area that it will bend those fibers. Okay, so let's put this all together and try to understand how those fibers will bend in response to different sound waves. By the way, before we start, a human being can hear frequencies between 20 to 20K. 20 is the lowest, 20K is the highest. So let's assume that someone is talking or we are listening to something and that thing is giving frequencies of 100,000 or 1000 hertz. Okay, and let's assume that 1000 hertz we will hear it in this region. What does it mean that we will hear it in this region? It means that those fibers will bend only to vibrations resulted from 1000 hertz. Those will bend to vibrations maybe resulted from 10,000 hertz. Those here, maybe they will bend to vibrations resulted from, let's say, 500 hertz. Those will bend to vibrations resulted from 20 hertz, right? So every time we have a sound that is creating vibrations in this fluid, at one point this sound will be able to bend part of the membrane depending on the pitches of the sound. Okay, for someone is talking, for example, and again we are saying that this person is giving us 1000 hertz. So when this sound wave coming to this area, it will cause it to bend. It will bend the membrane. It will bend the membrane. And this bending will push the fluid here, will push the fluid here. So it's kind of are pushing the fluid down. This will create a vibration in the basilar membrane. This will create vibration in the basilar membrane. Also, to make sure that we understand those vibrations are coming from where, we need to go back to the ear. So those vibrations are coming through the canal. I'll erase actually all of those lines. So vibrations are coming through the air canal. The sound wave is coming through the air canal, will cause vibration in the tympanic membrane to the malleus, to the incus, to the stapes. The stapes will push against the wall or the oval window, the bone, the temporal bone, and it will push the fluid in the cochlea, right? So it will push the fluids in the cochlea. So it will create a wave in the fluids of the cochlea. This is the wave we're talking about here, right? And this wave, based on the frequency of the sound, we will have waves that are like this, will significantly disturb the fluid, whereas we will have waves, if the sound is super low, we will have waves like this, super low. Okay, so the high frequencies will be capable to bend the membrane earlier, whereas the low frequencies will be capable to bend the membrane later toward the tip. So every time we are hearing a sound, we would remember that there is a vibration in our cochlea that causes bending in those fibers, right? So now we have a bend in this membrane as well, in the basilar membrane. We have a bend in the basilar membrane, So every time we have a bend in the vestibular membrane, think of it, we are disturbing this fluid inside, and we will cause the basilar membrane also to bend. Now, when the basilar membrane bends, it has to come back. Right? It's bouncy, so it will bend and it will come back. It will bend and it will bounce back. Now, what is above the basilar membrane? What do we have above the basilar membrane? We have the organ of corticels. We have those cells. Those cells, in response to vibration happening now beneath them, they will also start to vibrate. I need you to think of a boat in a lake. So if there was a lake or in the sea, like a boat, this is a boat, and now those are the waves. So as the waves are moving, they are going to move the boat up and down, right? Along with the wave. So that's exactly what's going to happen with the organ of corticels. As the waves here are moving the basilar membrane, those cells also will move. And they will move up. Those cells, we said they have their hair, and the hair is touching what? Or laying against the tectorial membrane. So when those cells are raised up, draw them again, this is the tectorial membrane, and those cells are raised up, what's going to happen to their hair? The hair will bend. So instead of standing like it was here, standing, it will bend up, it will bend. This bending opens the ion gates and induce action potential. Okay, so the whole concept is that we want those hairs to be bent and open the channels so we can induce action potential. How do I get them bent? I need to move them against the tectorial membrane. And how do I move them against the tectorial membrane? By inducing vibrations in the basilar membrane. Those vibrations would be a result of vibrations in the vestibular membrane. And those vibrations in the vestibular membrane are responding to certain frequencies. So if it's high frequency, we will hear those vibrations earlier, or the vibrations will bend the fibers earlier. If those are low frequencies, those vibrations will bend the fibers later, or toward the tip of the cochlea. So the brain will analyze those action potentials are coming from the bottom of the cochlea. So that's maybe a high frequency action potential. Whereas the action potential coming from toward the tip of the cochlea, the brain will analyze them as low frequencies. And that's how we understand that this is high sound or low sound. Okay, so that is how the cochlea is converting the vibrations into electrical signal. See on this drawing how the process is happening. So we have here again the oval window. Vibrations are coming, or sound waves are coming, converted to vibrations. So now we have vibrations in the sacral vestibuli. Based on those vibrations, if the frequencies are high, those will be able to bend the basilar membrane earlier at the bottom of the cochlea. But if they are low, they will travel all the way, and they will bend the fibers at the tip of the cochlea. Think of them, they are weak. That's why they cannot bend those hairs. So those hairs, they think of it, although it's not like really accurate, they have their own resistance. So it needs at the base, they need like really high power to bend them, whereas at the tip, they need low power to tip them. Okay, so the high frequencies would have more energy per se, and this will cause them to bend here, right? So that is how this process is happening. Now you might ask, what about this wave that will go and would come back, right? Because we said like the sacral vestibuli and the sacral tympani are continuous, so there should be like a wave that would come and go, and technically that happens, right? This wave might go all the way and it will come back and it will exit through the round window. This is something we did not mention before. The round window is not also a window, it's just like a spot on the bone that will absorb the vibrations, or yes, absorb the wave or the vibration. Think of it like a sponge. Okay, so the point is, or the idea is, that those vibrations or this wave in the fluid, we don't want it to hit and go back, right? I don't want this to happen. So the round window will act as a sponge that will absorb those waves and prevent them from going back. So that is how the sound wave is converted to action potential. Now the last few things in this in the ear are the vestibule. The vestibule, we said, along with the semicircular duct, those are responsible for balance. The vestibule is two chambers, continuous, utricle, and saccule. Both of them, they are responsible for the linear acceleration and the gravity presence. So what I need you to understand, that also we have hair cells there. Those hair cells surrounded by a fluid. And when we move our head, when we tilt our head, okay, the hair will bend, and that would induce action potential. So the vestibule is responsible for this linear acceleration, moving forward, for example, or backward, and also the head movement, bending it backward and forward, and also altitude. If we go in the elevator, for example, up or down, we understand that through the vestibule. The semicircular ducts, at their ends, there is an area, there is an expansion called the ampulla. So within the ampulla also we have a group of hair cells. We have hair cells also surrounded by fluid. When we move our head, when we rotate our head, the fluid would pass through those semicircular ducts and will cause the hair to bend. And that bending will be reported, will open the channels and will be reported as action potential. So the crista ambularis, this is the structure in the ampulla of the semicircular ducts. It's called the crista ambularis here. It's responsible for detecting the rotational movement of the head. To detect the rotational movement of the head. So that's it for this chapter. Let me know if you have any questions.